Braintime. Brain development during adolescence. Adolescence is a formative period for social, emotional and cognitive functioning. During adolescence, the brain undergoes structural and functional changes that may contribute to cognitive control, impulse regulation and socio-emotional processes, not only during adolescence, but also later in life. Neuroimaging can help us to get a better understanding of the mechanisms underlying behavioral change in adolescence, especially when it's being used to track longitudinal changes in brain and behavior, which is what we did in the Braintime study. While growing up, I have experienced that some adolescents do not have the same opportunities as others. I've always been curious how differences in the social environment interact with brain development. My goal is to get a better understanding of differences between people in how their brain processes reward, how this is related to one's social environment when growing up, and how this might influence well-being later in life. In the Braintime study, we investigated developmental changes in childhood, adolescence and emerging adulthood in 3 domains: cognitive control, impulse regulation and socio-emotional functioning. Starting in 2011, we for the first time combined different MRI modalities such as functional MRI, structural MRI and DTI with a wide range of behavioral tasks and other measures in one large longitudinal study. This allowed us to measure the relative development and interrelations of cognition, emotion and the brain. Braintime is a unique 12-year longitudinal study that combines hormone data, neuroimaging, behavior experiments and questionnaires to study developmental changes from childhood to adolescence and eventually adulthood. To capture the dynamics of development, participants' ages range from 8 to 25 years when they first started. This allowed us to capture a broader age range while also following individuals across development. This study started in 2011 and was one of the first ones to use this approach. The study consisted of 3 waves, separated by 2-year intervals, 2011, 2013 and 2015, in which participants were followed over time. At each wave, participants made a lab visit to the MRI scanner. Some participants were followed up with subsequent questionnaires to assess their development later in time. At all waves, we administered a cognitive control task to assess development of learning and strategies, and a personal and vicarious reward task to assess reward processing. At the 3rd wave, we also examined ambiguity in risk-taking. In addition, a wide behavioral battery examined impulse regulation, risk-taking, delay of gratification and impulsive aggression. All functions were examined in relation to structural brain development, gray matter density and white matter tracts, the social environment and the role of gonadal hormones. Using multi-level models of change, we test changes in developmental trajectories over time. In addition, we examine how development related to brain structure and function predict individual differences in future well-being. We already discovered that regions within the prefrontal cortex develop rapidly during the teenage years, which was related to more efficient learning strategies. At the same time, the reward region in the brain, the ventral striatum, is more responsive to rewards in mid-adolescence, also when you receive these rewards for close friends. But how do these findings relate to long-term outcomes? As all of the participants are now young adults, we are currently preparing an additional wave in 2022 and 2023 to assess their outcomes in adulthood. The upcoming wave will start off with an online survey, after which a subset of the sample will be invited for a 4th lab visit at the MRI scanner. We developed a new well-being paradigm in co-creation with adolescents and young adults to assess the relation between neurodevelopmental trajectories and well-being later in life. I hope to get a better understanding of the underlying mechanisms of well-being during adolescence and young adulthood. For example, can we predict which individuals will have a better well-being by examining the interplay between environment and brain development? Of course, we would love to share the results with you. Interested in collaborating on the Braintime project or using the data? please reach out to us. You can find the link to the metadata file describing all our measures here. This study was performed with a team of PhD students, postdocs and collaborators.